Hello, my name is Emily Sharif, and I'll be presenting my DNA microarray experiment today. The purpose of my experiment was to utilize the DNA microarray to test which gene mutation has occurred in our patient, precisely the BRCA1 and BRCA2 gene mutation. A DNA microarray test for BRCA1 and BRCA2 is used within our experiment. This is an effective way to identify mutations linked to an increased risk of acquiring these cancers by concurrently assessing thousands of genetic variations across the BRCA1 and BRCA2 genes. So, my research consists of determining BRCA1 and BRCA2 gene mutations. This has become essential for determining the risk of ovarian and breast cancer in families with inherited breast and ovarian cancer. For high throughput screening of genetic variants, including BRCA mutations, DNA microarray technology has proven to be an effective tool. An overview of DNA microarray technology's guiding principles and how it is used to find BRCA mutations is given in this abstract. Advantages of DNA microarrays are discussed, including their high sensitivity and specificity, as well as their capacity to analyze many genetic variants at once. BRCA1 and BRCA2 gene mutations have been affecting multiple families within our society. Cancers are rising on a daily basis, and acquiring this gene and making sure we can detect a mutation within the gene through DNA microarrays is very essential to helping people survive this cancer. The materials used in my experiment was a microarray card, which is the card that we will be determining whether or not the genetic mutation has occurred on, the quick strip of patient cDNA samples, equilibration buffer, hybridization buffer, control cDNA, micropipetter, disposable micropipette tips, handheld UV light, safety goggles, and latex exam gloves. Our procedure consisted of using the materials, taking the microarray card and putting the cDNA samples on them. We then put on the equilibration buffer and hybridization buffer, and then we put on the cDNA, and then we put that under the UV light and watch the results occur. And for our results, we have that patient 3 has the normal genes as their gene 9 is lit as yellow. Yellow means that the patient has expressed both normal cells as well as cancerous cells, meaning that they're at risk for a mutation, which we would need to look further into. And the other color is red. Red means that they're strongly expressed as just cancerous cells. And then we also have green, which is just normal cells expressed in the mutation. Well, sorry, not mutation, but within the cell. The other patients on the card have gene 9 as red, which means that the gene was strongly expressed in cancer cells. So this would mean that our patient would have to be looked out for, with further testing and genetic testing in order to figure out if this mutation has occurred. Verifying that this mutation has occurred will allow us to then make sure that this patient is coming in annually, to make sure that they don't have the cancer. And from there, we can make sure that our patient is healthy and safe. So to sum up our study using DNA microarray, technology has shed light on the complex landscape of genetic expression in cancer cells. Thousands of genes can be analyzed simultaneously with DNA microarrays, a potent technique in molecular biology that offers a thorough picture of gene expression patterns. With the aid of this technology, scientists can learn more about the molecular processes that underlie a number of illnesses, including cancer. We found a strong difference in gene expression amongst cancer patients through our investigation. Notably, according to the gene cards, one patient had normal gene expression, whereas the other instances showed clear mutations. The detected mutation indicates that the gene has been implicated expressed strongly in cancer cells, indicating that it may play a significant part in the oncogenic process, meaning that our patient has a high chance of getting cancer in the future. Therefore, we would have to monitor them throughout the years coming forth. Thank you so much for listening to my presentation today and taking the time out of your day.